Hello everyone, myself Neha Mesuria and I am going to talk about the PCR reaction, is also known as the polymerase chain reaction. Now, before directly jumping to the PCR reaction, I would like to summarize the basic replication mechanism of the living cell. Now, during the replication in vivo, what actually happens is the double-stranded DNA, those are the complementary sequences, both the strand will get separated and which will create the replication fork. These are called as the template sequences. Now, at the two template sequences are also called as the parent strands. The two different primers are attached and the extension of primer will take place with the help of the nucleotides, and the two different strands will be extended and finally get replicated, and from the template strand or from the parent strand, two new daughter strands will be generated with the help of enzyme known as the DNA polymerase. So this is what the basic of the polymerization reaction is, the replication reaction is. Now, using this technique, using this basic fundamental, Kary-Mullis is a scientist who first time invented the instrument called as the thermocycler and replicated as well as generated the n number of copies of the DNA. For that, the Kary-Mullis has also got the Nobel Prize. Now, let us talk about the PCR reaction and its principle. The PCR reaction uses the general principle of the replication, but this technique is purely in vitro technique. Now, in a test tube, this polymerization reaction will take place and n number of copies of the DNA will be synthesized in a small test tube. So, let us have an idea of the PCR reaction. So basically it has a 3 step. The first step is called as the denaturation step. Second step of the PCR reaction is called as the annealing step. And the third step of the PCR reaction is called as the extension step. Now do remember that each and every step of the PCR reaction has the different temperatures. And based on the temperature, this whole instrument work and the whole reaction work. So temperature plays an important role during the PCR reaction, right? So let us talk about the first step called as the denaturation step. Now denaturation simply means the separation of the DNA strands. DNA will always be in a double strand form. You have to separate both the strands by breaking their hydrogen bonds. So by applying the high temperature, that is say for around 90 to 95 degrees centigrade, you can separate both the strand and the two template strands will be separated. This is called as the denaturation step, the first step. In the second step, the temperature will be lowered down because there will be an annealing of the primer sequences. Primer sequences are those sequences are the short oligonucleotide which will initiate the replication of the DNA, or you can say that the synthesis of the new strand at the template strand. So the primer sequences in both the direction, in forward direction as well as in the reverse direction, or you can call it as a forward primer or the reverse primer, will be attached at the two template strands. This step is called as the annealing step, or annealing simply means the attachment. Attachment of what? Attachment of the primer sequences. This is the second step of the PCR reaction. Now in the third step of the PCR reaction, which is called as the extension step. Here the machine is extending the primer sequence because at the annealing you cannot stop the reaction, right? Reaction is not basically stopped, it is still ongoing. You have to extend the sequence in order to generate the complete sequence. So, after the primer sequence get attached to the template strand, one by one the nucleotides get attached to the primer sequence and the sequence will get attached and will lengthen and get replicated. So, this is called as the extension of the template sequences and complete strand will be synthesized in both the direction. Forward, that is from 3' to 5' direction, and in the reverse direction, that is again the reverse sequence 3' to 5', right? The polymerization will always take place in 3' to 5' direction. that this you have to remember, right? Now this whole reaction, the polymerization reaction, or you can say that extension step, will take place with the help of enzyme, the most important enzyme of the replication, called as the DNA polymerase enzyme. This enzyme can work at the higher temperature of the thermocycler instrument, and that is why this enzyme must be a thermostable enzyme. Now you must be knowing that the enzyme which are working in the living cell, which are not thermostable, they are thermolabile enzymes, thermosensitive enzymes. So for the enzyme to work at the higher temperature of the thermocycler instrument, you have to isolate the enzyme, you have to separate out the enzyme from the bacteria which can live at the higher temperature, are also called as the thermophiles, right? So you have to take the enzyme from the bacteria known as Thermus aquaticus, which can survive at the higher temperature, and that is why that enzymes can also work at the higher temperature. So for the PCR reaction, is the enzyme Taq polymerase is used, and this Taq polymerase is taken from the bacteria known as Thermus aquaticus. So this is how the template strands or the parent strands has got replicated in a single test tube, in a small test tube in vitro, and this is how n number of copy. Now if I repeat whole reaction, the denaturation, the extension and the annealing, if these all steps are replicated, you can generate the n number of copies of the DNA, and you can utilize this technique in the research, in the identification of the various bacterial as well as various viral species. You can also identify certain genetic disease, say for example genetically occurred cancer, right? So this is the technique which is very useful in the biotechnology field, and that is why it is very much used technique. Whenever you go in research, especially in living cell research, at that time this technique is definitely been used. So thank you so much. I hope that you have liked my lecture. Thank you so much.